want to say good afternoon now that it's after 12, and thank you for joining us today for the fourth lecture in the University of Regina's 2021 Research with Impact series. I'm Lisa Mitchell, the Associate Vice President of University Advancement and Communications at the University of Regina, and it's my pleasure to serve as the host of today's lecture. Over the next hour, you'll hear from Dr. Andrew Cameron, a leading molecular microbiologist with a specialty in infectious diseases, a timely topic. Dr. Cameron has been an incredible resource for the University of Regina's executive team during the past 17 months. He's kept us informed and provided us critical advice as we made operating decisions during the pandemic. Before I introduce Dr. Cameron, though, I'd like to start by acknowledging that today we're gathered virtually on Treaty 4 and Treaty 6 territories, which are the ancestral lands of the Cree, the Saulteaux, Dakota, Lakota, and Nakota's peoples, and the homeland of the Métis, on whose territories the University of Regina resides. This is an incredibly important part of our collective past, but also an equally important acknowledgement for our collective future. The University of Regina's alumni department, together with the Lifelong Learning Center, is pleased to be able to offer the Research with Impact series virtually. I had the pleasure of hosting several of these lectures in 2020, but this is my first time hosting this lecture in this series this year, and I'm looking forward to hearing Dr. Cameron speak today. I hope that your time with us inspires you to come back for future sessions. We do have more this year. And an interesting and positive outcome of COVID was that it pushed us to move this series to a virtual format. And this has enabled us to reach alumni and friends from beyond Regina, across North America and around the globe. And I want to welcome everyone attending today, but a special welcome and a hello to those of you who are attending from outside the city of Regina. We are so glad to have you as a part of the audience and have all of you here. Just a few housekeeping items before we begin. These sessions are being recorded and will be posted on the UofR alumni website so that you can watch the presentations again or share them with others if you'd like. You'll see the link to the alumni website in the chat function, which is at the bottom of your screen. It's a nice little bubble that looks like the bubble that you'd see in a cartoon when someone's talking. And that has the link if you'd like to access the video later. And finally, we do aim to have today's session done by 1 p.m. Dr. Cameron will present and we'll try to be done his presentation about 12:45, which will allow for 15 minutes of discussion and questions from the audience. And so now it's my great pleasure to introduce our fourth speaker in the University of Regina's 2021 Research with Impact series, Dr. Andrew Cameron. Thank you for joining us, Andrew. Dr. Cameron he's an associate professor of in the Department of Biology and the co-director of the Institute for Microbial Systems and Society at the University of Regina. That's a big mouthful, Dr. Cameron. Dr. Cameron's research examines the relationships between humans, animals, and landscapes that contribute to the emergence of new infectious diseases. His lab leads and participates in multiple COVID projects in partnership with provincial and national public health laboratories in Canada. They're working to develop improved genetic tools to detect viruses and identify variants, as well as understand how viruses jump from animals to humans. And so, as I mentioned earlier, Dr. Cameron and his work has been a great resource to the university leadership over the past few months. Dr. Cameron has been instrumental in leading our response to the COVID-19 pandemic and our long-term pandemic-related planning, and he was chair of the university's recovery planning group. Today, Dr. Cameron will talk about public health efforts to track and stop the pandemic, current vaccine technologies, and the implications of emerging viral variants for vaccine effectiveness and infection risk. He will also describe his lab's COVID-19 research at the UofR as they work to detect viruses and bacteria in COVID-infected patients to understand the consequences of co-infection. Following his presentation, as I mentioned, we'll have about 15 minutes for a question and answer session, so get your questions ready. And a colleague, Hertha from the Lifelong Learning Center, will be moderating that question and answer session. As you listen to Dr. Cameron's presentation, please put your questions in the chat feature. Again, that button that looks like a bubble to talk in a cartoon, which you'll find at the bottom of your screen. And then Hertha will share those with Dr. Cameron shortly after his presentation ends, and we'll do our best to get through as many of those questions as possible and as time allows, with the intention again of having you free by 1 p.m. And if you're comfortable at home, feel free to have your lunch or a coffee or whatever, but perhaps during the presentation, we could ask you to just close your video to allow Dr. Cameron to focus on presenting his presentation. So now I will turn it over to Dr. Cameron. Dr. Cameron, the floor is yours.
Thank you very much for that introduction, and thank you very much for having me to this speaker series. If I do everything correctly today, it will be to stimulate, I think, a lot of our collective interest in this big topic of COVID and infectious diseases. And I'll do my best to leave as much of 15 minutes and hopefully more for us to have that discussion. So I've prepared a few slides, and I've left out vaccines by and large from my presentation material today, just to focus a bit more on the types of research that we do at the UofR to understand, at least in my lab, to understand COVID and the pandemic. And that's a very genetic basis, which I'll walk you through in today's presentation. And then how that helps us understand variants of concern. So I'll talk a little bit about Delta and the other variants that we're hearing about. Show you how we can use these technologies to discover new viruses, including novel coronaviruses. So we know very, very little about all the coronaviruses that actually circulate in the world. And then I'll wrap it up and move to discussion. And then if there is interest and people have particular interest in discussing the vaccines, I'm prepared to talk more specifically about the vaccines, but I won't present on them presently. Okay, so I'm going to share my slides with you. Okay, so as Lisa introduced, I'm both an associate professor in the Department of Biology at the University of Regina, and I've been at the university for 9 years now. And in that time, about early 2017, along with a colleague in biology, Professor Christopher Yost, we founded the Institute for Microbial Systems and Society, which is housed within the Faculty of Science. And this material that I'm going to present to you is really conducted within that institute, because that institute thinks big and broad, and it's through that institute that we've brought together many of the technologies that we use to understand the genetics of COVID and other infectious diseases. There's my contact info that you can see there, and also can find that on the University of Regina website. If you have any follow-up questions, I will do my best to reply via email, but please be patient and send me reminders so that I can get back to you within a few days. So broadly, as you heard in that introduction, much of my interest in microbes is partly as infectious diseases and partly really as the ecology of all these microbial entities, bacteria, fungus, and viruses that are everywhere in the world around us, but we can't see. So we really live on their shoulders. They cycle the nutrients around us. They make it possible for us to live. They make much of the air we breathe. But they also, some of them can cause disease. So I'm very interested in this relationship between humans and animals in different landscapes, both agricultural or wild, and then the terrestrial and aquatic systems around us. And we call this type of relationship, and trying to understand how pathogens can move between these different environments, that now sort of falls under the phrase One Health. And so I'm, I guess, increasingly becoming a One Health biologist as I work to understand these relationships. And the tail end of my talk today, I hope I'll bring it back to that One Health understanding. And at the University of Regina, I really got started using genomics to understand infectious diseases in about 2014. In 2015, started working with what was then the Saskatchewan Disease Control Lab to sequence bacterial pathogens in the province of Saskatchewan. And then shortly thereafter, I had the opportunity to travel to Western Africa, to Sierra Leone, to try to use our DNA sequencing technologies that we had at the University of Regina to discover new infectious diseases and to develop better diagnostics so that we can go to anywhere in the world and provide rapid detection of diseases and characterization of those diseases. And that has now grown into multiple partnerships across Western, Central, and Southern Africa. And I'll be telling you a bit about Central African coronaviruses today. And it's also expanded within Saskatchewan as well. So now we have a large project looking at antimicrobial resistance and the evolution of pathogens in agricultural systems, and also understanding different diseases that infect not just humans, but also livestock as well. My lab is also involved in a Western Canadian effort to understand tick-borne diseases. So everything from the forests of British Columbia that you see there to the prairies in Saskatchewan, and the different diseases that ticks can carry and transmit from animals to humans. So in my lab, what do we do to try to address these different infectious diseases? Well, I like to break it down into about 4 different ways that we study infectious disease. And in the top left-hand corner there, you see where we use molecular epidemiology, and that's what today's talk is going to be about, which is where we can sequence the genetic material and then use genes and their sequences to reconstruct both where infectious diseases come from and then how they're transmitting either locally within our communities or around the world. We also take an ecological approach where we'll study pathogens and how they interact with different microorganisms in their environment or different aspects of their environment. I won't talk about that today. In the bottom right-hand corner, you can see we do some molecular work where we zero in to understand how, in that case, bacteria can transmit genes, so how bacteria can acquire things like antimicrobial resistance from them. Again, I will not talk about that today. And then in the bottom left-hand corner there, one thing that we're very into is discovering new genes. So for example, when we find antimicrobial resistant pathogens, we need to figure out how did they become resistant to antibiotics. And we can do that using the DNA sequencing technologies that I'll describe today. But I will not, again, today talk about that aspect of gene and function discovery. So today we'll focus just on this molecular epidemiology and how we use it to understand the COVID pandemic. And so here's a photo from our institute in pre-COVID times. And I want to specifically acknowledge members of the Institute who have generated the data that I will get to present today. So these are the excellent colleagues who actually do the research in the laboratory space. And this, the main COVID project of my lab has had 2 project leads, and now we're on to a 3rd, in part because the members of the lab are experts in the use of genetics and genomics to understand infectious disease, and who needs them is our public health laboratory. So very recently, both Dr. Keith McKenzie and Dr. Kara Luce, that you see circled there in pink, have moved from working in our institute to working with the Roy Romano Provincial Lab, doing exactly the same thing of sequencing genomes to understand the COVID pandemic. And circled in yellow there are Danae Sukin and Dr. Jen Russell and Ashton Size, who are all members of the Institute who are currently working on this COVID project. And then in orange is Kevin Kuczynski at the BC CDC, who has come out to work with us at IMSS and has been instrumental in us developing genetic technologies to understand COVID. So here's a graphic showing really the pandemic roller coaster that we've been on. We've all lived it. I don't need to say, I think, too much about it. We're in a current phase that is very positive in Canada, where infection rates are continuing to fall and vaccination rates are climbing. They're not climbing as fast as we would like, but again, I won't talk too much about vaccinations specifically today. But we're certainly not out of the woods and we're not away from this challenge yet. And one thing we do continue to hear about are these variants of concern that are spreading within Canada and within our province. And then if we look globally, so this graph on the bottom is showing us that, again, we're not out of the woods yet with this pandemic, that unfortunately infection rates are now increasing globally. So you can see from a high in April of this year, global rates of new infections, which is plotted there for each day, were falling until about mid-June, and now are starting to climb again. And there's now a global average of about 500,000 new infections detected each day around the world, and that number continues to climb. So what's happening with COVID? Well, one of the big problems is that the virus, when it jumped from an animal into humans, which I'll touch a little bit more on, is that we were not the perfect host for it. And it was able to infect us and hence start this pandemic. But what's been happening is as it continues to spread, which is primarily through an infected individual seen there in red, exhaling or coughing out many viral particles shown as little red dots, is that a susceptible uninfected individual nearby can inhale those viruses, and those viruses can then infect the upper respiratory tract of that second individual, causing a new infection. Now, as the virus continues to spread, it has to replicate, make more of those red dots, and as it does so, it can mutate. And mutation is a constant feature of viruses, and viruses like coronavirus actually mutate quite quickly, and I'll show you that on the next slide. So there you can see that in that newly infected individual, one of those red dots has just changed to blue, and that's my illustration of a new variant evolving in that transmission. And now as that newly infected individual coughs out more virus, they might then spread that variant to the next person, and the virus will continue to mutate as it spreads. So we do know that SARS-CoV-2, the virus that causes COVID, did jump from an animal into a human. There is a possibility it went via a lab along the way, but I won't talk about that today. But we do know that these viruses originate from animals, most likely bats. Bats contain most of the coronavirus diversity on Earth, and certainly contain viruses that are very, very closely related to the SARS-CoV-2 that is spreading in humans. And we've certainly seen in this pandemic that humans can give the virus essentially back to animals, and infections do happen in other mammals from humans with this virus. So it can jump between animal hosts. And what's happened since that very first virus jumped into a human sometime in the latter half of 2019 is that, as I just illustrated on the previous slide, as time has gone on, the virus has continued to mutate. And so if we look at the number of mutations that are accumulating in the virus over time, what we see is this growth in mutations. And each dot there illustrates a different SARS-CoV-2 virus isolated from a different person somewhere in the world, and the colored dots indicate where in the world that virus was isolated. And then all the genetic material is sequenced, and we can identify the mutations. And so you can see that now on the right-hand most of the graph there, that all of the viruses that are detected around the world have about on average 40 mutations that differentiate them from the original virus that first jumped into a human back in 2019. And in fact, that original virus that jumped into humans is not detected anymore. It may have gone extinct in early 2020 because we don't see it anymore. It's been replaced by these mutant variants. Now, most of those mutations do not matter to the biology of the virus. They are probably neutral. They do matter very much to a person like myself, a microbial geneticist, because they're the material that we use to track and understand the evolution, and importantly, how SARS-CoV-2 continues to evolve to adapt to us as its preferred host. So one of the reasons we're seeing more infectious variants evolving now is because we were not the perfect host for SARS-CoV-2 at the beginning, and it's evolving to better use us as a host. So it's becoming more transmissible, better able to replicate in our respiratory tract. So there are key mutations that probably contribute to those new features. And one of our tasks is to figure out what those mutations are and sort them out from all these now thousands and thousands and thousands of other mutations that are accumulating in the viral genomes as they spread and diversify around the world. So to sort of wrap up that transmission illustration before I jump a little bit more into the molecular biology. You can see on the left-hand side there, you might now see the term wild type SARS-CoV-2 in the news media, and that refers to these non-variants of concern form. So a virus that has essentially that original transmission rate and facility that we saw through 2019 and early 2020. Now, these new variants of concern, which are taking over, certainly in some countries, we see like in the United Kingdom, where Delta has now taken over, completely displaced an earlier variant of concern called Alpha. One of the reasons they're able to do this is that, as I just mentioned, these new variants can replicate to higher numbers. So each infected individual can be expelling more viral particles into their environment. And importantly, a susceptible individual might only need to inhale fewer viruses because it's better now at binding and perhaps evading and escaping immune detection right away and replicating faster when it gets into that new host. So infection rates can go up because the virus can transmit better between individuals. Okay, so now, a little note that the race is really on, because as long as the virus continues to spread like this, it's going to continue to mutate, it's going to continue to evolve to use us as a host. And really the best tool we have to fight this evolution and ongoing pandemic is vaccinations. And vaccinations can stop these variants. So we just need to get those vaccination rates as high globally, both within Saskatchewan and around the world, as fast as possible. Okay, so now I'm going to zero in to tell you a little bit more about the molecular biology of the virus and how we think about it and how we understand how it's evolving, and then how we use those mutations to track the virus and detect new variants, and then hopefully be able to identify when new variants of concern evolve and really mitigate. So that's the ideal, is the faster we can detect the virus and variants, the faster we can stop the transmission and ultimately stop this terrible pandemic. So there in the middle of your screen, you see a cartoon, essentially, representation of what the virus looks like. And this is, of course, expanded greatly for us to see. These things are so small, and you may have heard this number, but if we took all of the SARS-CoV-2 viruses on Earth at the moment, every viral particle, and put them all together in one place, they would fit in a can of soda or a can of Coca-Cola. So they are that small that we could take those many billions of viral particles, and all of them on Earth causing this pandemic at the moment would fit in a single can of soda. So they're very small. So we're looking at a greatly expanded picture here. And the piece that we hear about the most, and the one that is very important for both the vaccine and understanding the evolution of the virus, is that spike protein. And that's the red globule that you see sticking on the outside of the virus. And why that's so important is that protein is what allows the virus, so here's the spike protein, and here's our tissue. It allows the virus to interact with our tissues, bind to our tissues, and then be engulfed by our cells so that it can enter one of our cells and start to replicate. Now, all of the vaccines are based on that spike protein. So when we receive the vaccine, our body is being given the instructions on how to make that spike protein. We'll have vaccines that come that will also just be the raw spike protein used in the vaccine. And then once that spike protein is inside, say, the muscles of your arm, we're able to start to build both antibodies and cellular defenses against those proteins, which can help our body then block further infections or any infection by this virus. So if we zoom in to look at one of these spike proteins and now look at the mutations that can arise there, this is a graphic from a while ago now, but I left an old graphic up because it does a nice job of showing how, as the virus has been spreading around the world, it has been accumulating many mutations at different positions in this spike protein. Some of those are probably quite important for the increases in transmissibility and the ability of the virus to evade our immune systems, because our immune systems target that protein. A few of those mutations, those variants, we've heard a lot about because they defined essentially the early variants of concern. The N501Y was the first one that really caught global attention because it's characteristic of that alpha variant that arose first in the United Kingdom. And that E484K, so that showed up in South Africa in what is now called the beta variant and also in the gamma variant. And they both also share that N501Y. So it's thought that that 501Y mutation can assist the virus in binding to our tissues, and that 484K mutation might help it avoid our immune system and immune detection. Now, those aren't the only mutations in the spike protein. So we still don't understand, there's so much unknown about these viruses and those different mutations, but it does help us to have a few to focus on. And I'll show you why also from a diagnostic perspective on the next slide. And there in the bottom right-hand corner, I've copied the list of mutations found just in the spike gene, or actually changes in the spike protein that are characteristic of the Delta variant. And you can see that that's 11 plus different changes that are found in different Delta variants around the world. So a lot of mutation has happened in these viruses as they continue to spread. Okay, so now I'm going to tell us a bit about the viral genome and how we use genetics to detect the virus, and then the following slides, what we're doing in our institute to better understand the evolution of the virus. So there you see illustrated in that bluish color is the whole genome, and a genome refers to all the genetic material in a biological entity. So in this case, that's the SARS-CoV-2 genome, and you can see that it's about 29,900 bases long. And each of those different little blue arrows represents a gene. And a gene means different things to different people, especially when we're talking about viruses. But that one called S, that's the spike protein, the gene that encodes the spike protein that we're so particularly concerned with. Now, if a person goes for a test to see whether they've contracted the virus, in Saskatchewan and around the world, the gold standard, the preferred test, is a PCR diagnostic test to detect whether the virus is in a person's respiratory tract. And PCR diagnostics has many, many benefits, and that's why it is our primary test, both here and around the world. It's fast, it's accurate, there's very low error rates, so very low false positive, very low false negative rates, and it detects the presence of the virus. So the PCR test is detecting whether the genetic material of the virus is present in that person's respiratory tract at the time of the test. Now, one of its shortcomings, though, is that we have to know what we're looking for. So PCR is based on this ability to target little pieces of the genome, and those are highlighted in that orange and red color there, that currently in the Roy Romano Provincial Laboratory, they will test for part of this gene 1AB, and they'll test for part of the spike protein. And if either one of those is positive in the PCR test, then that person is presumed to have the SARS-CoV-2 virus because the genetic material was detected. Now, thanks to their cunning and ability to continue to improve this diagnostic test, in Saskatchewan, they've added different targets as well that allow, with PCR, to look for specific mutations that can be representative of the variants of concern. And this helps because now they can use that very fast diagnostic that's very accurate, very reliable, highly sensitive to look for whether there's some mutations that are characteristic of, say, alpha or other variants. Now, unfortunately, for all those benefits of PCR, it can only do so much. So it's limited in how many additional mutations essentially that it can detect. So it does not tell you all those many mutations that are in the spike protein that are, or across the whole genome, that are required to say, aha, this is the Delta variant, but it can very quickly, within 24 hours, give public health a very good estimate of, say, how many alpha variants are circulating in Saskatchewan at the moment. But to really understand the variant, what we need is whole genome sequencing. Now, unfortunately, it's slower. It currently takes about a week or up to 2 weeks at the Roy Romano lab to be able to sequence the genome. And it's not as high throughput. So only in Canada, it's about 1% of SARS-CoV-2 positive specimens now are actually having genome sequencing to look at what is the sequence of that SARS-CoV-2 isolate. So we're only seeing really a small piece of all the genetic variants in the SARS-CoV-2 are across Canada at the moment. And that's true for across the world as well. Now, the benefits of whole genome sequencing, it's very accurate. It has a very low error rate as well. It detects all the genetic differences. So now we're sequencing all of that blue sequence up here to see the whole genome of the virus. And what it also allows us to do is to find new species. So we can use it to find more than just SARS-CoV-2. And that's a key piece of our research. So in the next few slides, I'm going to now just wrap up by telling you about what we're doing specifically in our institute to use genomics to track SARS-CoV-2 and other viruses. But just by last sort of pitch of why doing the genomics, so sequencing all of that genetic material is so important, is that when we have all of that information and can see all of the bases that make up the virus in a specific person, in an infected individual, that allows us to now use very powerful comparative tools to reconstruct how the virus has been spreading. So we can identify transmission routes, as you can see on the right-hand side here. Early on in the pandemic, for example, when we first started sequencing SARS-CoV-2, we could tell where certain viruses had come from that had arrived in Saskatchewan, which parts of the world they had come from based on their genetic sequence. So we can find the sources. We can more specifically even find the animal sources, which is a very active area of research that we're involved in as well. And we can discover new diseases. So at the U of R, I'm going to tell you about a couple of the big questions that we're asking around SARS-CoV-2 and COVID infections at the moment. So what we're interested in is what happens if a person has COVID and they contract another infectious disease at the same time, or maybe they already had another infectious disease and then contracted SARS-CoV-2. So what are the consequences? And we already have a sense that it's not good. If your body has to fight both influenza virus and COVID at the same time, that's a very hard battle for your immune system and for your poor heart that has to pump more blood and all of your bodily functions that just have to work harder to fight multiple infectious diseases. What if it's COVID and an antibiotic resistant bacterium? We know almost nothing about that at the moment. So this is what our research hopes to address. And then also we want to use our tools to understand how many different coronaviruses actually exist in nature. We knew very little about the genetic diversity of coronaviruses before this pandemic, and still during this pandemic, almost all global attention is focused just on SARS-CoV-2, and very little work is being done to understand coronaviruses in general. So where's the next coronavirus going to come from? One thing we've learned from this pandemic is coronaviruses have probably been causing pandemics like this in humans for millennia. And we used to think that some of the big historical pandemics were caused by influenza primarily. Now that's being reevaluated, that some of those past very deadly pandemics have hallmarks of coronavirus pandemics. So if coronavirus is able to keep jumping into humans, how do we anticipate what are the next coronaviruses that could do that? And what is it that allows them to do that? So I need to wrap up here. I'm just going to show you some of our data. So a key piece of doing genomics is that it really is a problem of finding a needle in a haystack. So what you see on the left-hand side there is a nasopharyngeal swab, or it could be a swab from a bat rectum, anywhere that we want to take a little bit of biological specimen and find the viruses or bacteria or other infectious diseases. But finding the pathogen is hard because almost all of the biological material on that swab belongs to the host. So for a nasopharyngeal swab, if I just stuck it up my nose now, it would be mostly covered in my DNA and RNA. And it would also have a lot of bacterial DNA and RNA from all the different bacteria that live in my upper respiratory tract as well. And that's what we can illustrate as the haystack of all that genetic material that ultimately we don't care about because we want to find the specific causative agent of disease. And that truly is the needle in the haystack, because the amount of viral RNA in all of that total genetic material is very, very, very small. So when we face this problem that there's such great benefits of having that sequence, but it is a needle in a haystack, what do we do to find it. And that's where we've really invested much of our focus in our work at the University of Regina, is to really help advance this technique called probe capture. And very quickly, what it does is we extract all the genetic material from that swab. In that first gray box, you see we're converting the RNA, because SARS-CoV-2 stores its genetic material as RNA. We convert it to DNA, and then we do this probe capture. And very briefly, what probe capture is we develop these baits, and so you can see one highlighted in red there, that is allowed to essentially hybridize, is the term we use, to bind to the genetic material from the SARS-CoV-2 virus and then enrich it. So we can pull out those needles and then focus our sequencing effort just on the needles that we want to sequence and ignore all the rest of the hay. And then we can do that just on our benchtop sequencers. So these are DNA sequencing machines that we have at the University of Regina. Or if we want to get even more depth, we can contract it out to go for one of these bigger sequencers to get even more genetic sort of background and understand maybe even the human genetics if we wanted. And this is a very scalable, many advantages to this technique, but it's highly sensitive. And importantly, it allows us to discover new things that we didn't know existed before. So briefly, our results so far from doing this type of work in specimens, so from people in Saskatchewan, is that we generate excellent whole genome sequence of SARS-CoV-2. So we can, with those genomes, we can reconstruct different aspects of the outbreak, watch how the virus is spreading across the province, infer who was maybe transmitting it to who. Looking back at earlier specimens, we can find early variants long before our public health lab was able to do the DNA sequencing. But one thing we found, and so far we've done 75 specimens, is that we set up this technique to find co-infections, but we didn't identify any co-infections so far. But we have validated that the assay can detect other viruses. So for example, we've been able to do some whole genome sequencing of influenza viruses from Saskatchewan specimens. And what you see there is an illustration of the sequencing result from a piece of the influenza genome, and each of those colored bars represents a genetic novelty that's different from the reference genome. So we're able to see which variants of influenza very specifically are circulating in Saskatchewan. And that type of work is very rarely done because it takes so much extra effort. So this is great. We're taking a leap forward through this type of technology to better understand other infectious diseases in Saskatchewan. We also stumbled across a virus we had no idea circulated in respiratory tracts in Saskatchewan. Not only did we detect it, but we sequenced its whole genome, and that's what's illustrated on the bottom there. That's all of its genetic material. And again, the colorful bars show us the novelty, the genomic novelty or different sequences arising in that Saskatchewan isolate that we sequenced. And now to wrap up, I want to tell you about what we're doing to find new coronaviruses using exactly the same technology. So this is tricky because what we want to do is pull out, enrich things that we don't know what they are yet because we haven't sequenced them. And the whole ability to enrich that DNA, we need our probe to be able to bind to the DNA or RNA, depending on what type of specimen we're looking at. And what's so neat about this technology is that we only have to have a very approximate sense. And thanks to our understanding of genetic diversity and evolution, we can develop probes that can capture large sort of areas of evolutionary diversity that we just don't know yet, but we can predict exist. And that's exactly what we did with coronaviruses. So thanks to collaborations that I've been working with and setting up for years now with scientists who have been sampling bats across Central Africa, we received specimens from the Democratic Republic of Congo from coronavirus or from bats, different bat species there. These were very low quality genetic material. So this was part of the challenge is could we detect the viruses and could we actually sequence their whole genomes? And we use this probe capture technique to do that. And one quick thing I want to show you is how we really greatly improved this technology. And this was Kevin Kaczynski that I mentioned, our collaborator at the British Columbia Center for Disease Control, was instrumental. He developed the computational workflow to do this. And what you see in those orange dots is using the probe capture technology from 2019 to capture even just known coronavirus diversity. Those orange dots show us sort of what percentage of current known diversity we would expect to capture. And you can see that most of it is sort of 30% or lower. But then using Kevin's approach to design probes, we could actually develop a probe capture technique that caught 90% to 100% of what's currently known of coronavirus diversity. So that already gave us good hope that when we turn this to the unknown, we would be successful. And sure enough, we have been. This has been a very exciting project because we were able to detect the coronaviruses. We were able to generate genetic sequence from these different coronaviruses. And these are coronaviruses from across the wide diversity of coronaviruses. And what I'm showing here in these two illustrations is at the top is a reference sequence for a coronavirus. And then blue is mapping our genetic sequencing in our lab to those known coronaviruses at the top. And what you can see is very dramatic here is a big missing section. So we've sequenced these new novel coronaviruses with our probe capture technique, but we don't recover, or we don't appear to recover, the spike gene. And the key point here is that the green is a reference genome. So this is a previously published sequence from Central Africa. And the coronaviruses that we're sequencing just don't match at that spike gene. Now, this bottom red line does show us that we've actually sequenced the whole genome, including that spike. So what that means is not only have we found all the thousands of genetic differences across the whole genome, we've also been able to discover these wholly new spike genes. And for my final slide to wrap up, just last week, a paper was published showing a very similar type of analysis of coronavirus diversity in Southeast Asia. And at the top there is that SARS-CoV-2 genome, and then comparing these novel coronaviruses that were sequenced in Southeast Asia. And each one of these lines shows the degree to which the novel coronavirus genome sequence matches SARS-CoV-2. So you can see in green there, it's sort of between 75 and 85% similarity to SARS-CoV-2. And with all of these different coronaviruses, when we get to the spike gene, we can see that similarity plummets. And that's because the virus is being driven to evolve very quickly in the spike protein because our immune system targets it, and so does the bat host and any other mammalian host that a coronavirus will find itself in. It always has to evolve quickly in that spike gene to evade its host. And so using these whole genome technologies, and particularly this probe capture approach we're using, we're able to not only isolate those viruses from these even sometimes very poor sample quality and able to get that whole genome sequence, we're able even to sequence new spike genes and better understand how not just SARS-CoV-2, but all coronaviruses are evolving. And in that last bullet point there, ideally, we want to understand better how do these viruses jump between different animal hosts. Okay, and I will just finish there by thanking, of course, everyone in the Institute who has contributed to this work and our collaborators, particularly in public health, who have been instrumental in providing the guidance and the samples for us to work on, and the different funding agencies. And thank you to the University of Regina and to Lisa for inviting me to give this presentation. I will stop sharing.
Thank you, Dr. Cameron. Absolutely fascinating work. I'm glad it's you that's doing it because it's very scientific and very complicated, and you're making it sound very simple, but I know it's not very simple. So right now we've got Hertha with CCE, who is collecting the questions that you have through chat, and I'm just going to hand it over to Hertha to facilitate the question and answer.
Okay, thank you. I have a few questions, and they're really long. I have a couple of questions. In one of the slides, the person was shown as being unvaccinated and spreading the virus. My question is, I understand that being vaccinated, we are still able to spread the virus. Is this accurate? If yes, are we less likely to spread the virus or the variant? And this is from Janet.
Yes, well, I can address that specifically. So it's true that vaccination is very protective. And so most infection now is occurring, at least in Canada, in unvaccinated individuals. But it is certainly true that vaccinated people are not sterilized. So that means that the vaccine can protect us, and it certainly protects us from disease and death to a very great degree. It does not necessarily mean that you will not be able to transmit the virus. So why that happens is our immune system is able to mount great defenses that can keep the virus at bay and keep it from replicating and destroying our tissues to an extent that it can cause disease and death. But the virus can get lucky and it can get in there, sneak by the antibodies and some of those killer cells, replicate a little bit, and still be able to transmit to another person before the vaccinated individual, their body clears it or kills what little bit of virus got in. So what I'm trying to impress there too is that, yes, it's possible for vaccinated individuals to spread the virus, but they're much less likely to spread it because the virus cannot replicate in most vaccinated individuals. It can just not replicate to the same high numbers that help it be expelled in great clouds to transmit to other people. So anytime you're breathing out less of the virus, it's less transmissible. And vaccination really helps to reduce, even in the rare case of infection, it helps reduce that amount of virus that's being exhaled by an infected individual.
And her next question is around testing for the virus. Can you explain why the individual may have false negative tests, multiple negative tests following a known contact with someone with the virus? Is the 1AB or the S not identified for that individual? Is it that it has not yet entered their system, etc.? She said.
It really depends on the diagnostic test, and this is going to become ever more sort of complicated in what we see in Canada and around the world as more and more tests become certified and available. So currently, the PCR test is the standard test in Canada, and in most cases, it's the only sort of allowed test to determine whether a person is infected. And it is sensitive, but it's only so sensitive. So if a person, for example, is very early in the infection cycle, or also very late in the infection cycle when the number of viral particles is very low, as you described in your question, yes, it could be negative at maybe one of the targets or both of the targets, even though there's a very, very small amount of the viral genetic material there. For public health testing, if it comes up negative at one of the targets and positive at the other, then they'll actually rerun the assay to improve detection and confirm whether that person is in fact, or try to improve their confidence whether they are getting a true positive signal from that specimen. So the other thing to bear in mind is that the PCR test is testing specifically for the presence of the virus itself. And during infection, a person might be carrying a detectable amount of the virus for maybe 2 weeks or so. And they may have been sick and spread the virus, but by the time they get the test, their body has cleared it enough that it's no longer detectable. Now, many of the new diagnostics that are becoming available are going to be based on whether a person has antibodies against the virus. And that gives us a very different picture, because once a person develops antibodies against the virus or the vaccine, and in fact, some of these new diagnostics can discriminate between whether a person has made antibodies against the vaccine or a true infection of the virus, is that once a person is making the antibodies, we can detect those for many, many months after the infection. So an antibody test doesn't tell us whether the person's currently infected. It just tells us whether that person has been exposed or contracted, more specifically contracted the virus at some time in their past. And so we get different pictures from these different diagnostics. And I hope that addressed your question.
Okay, I got one from Caroline. You mentioned the virus can be transferred from humans to animals. Do we know if of any cases of family pets getting COVID-19?
Well, certainly there are cases of both cats and dogs contracting the SARS-CoV-2 virus. I don't believe there's any cases known of them developing the disease, which generally we refer to as COVID for the disease. So yes, we can transmit to other animals. It's very clear in the ferret and mink farms that humans can transmit to other mammals, the SARS-CoV-2 virus, and it has been detected in a few cases of companion animals like animals and cats, but I'm not sure that those pets have developed any noticeable disease or symptoms from those infections.
Okay, we got one from Gayleen. In terms of risk benefits, is there really a big benefit to healthy individuals being vaccinated when the risk of developing severe disease has been proven to be minimal, in contrast to the higher risk individuals that have been shown to be older and have comorbidities? They have more benefits of being vaccinated. What are your thoughts on having a more targeted approach to ensuring that higher risk individuals are vaccinated while those that are capable of fighting the virus in their own immune system and developing natural immunity are not?
So I think it is absolutely critical that everyone, every eligible adult get that vaccination, and youth and eventually children, because whereas it's true that rates of disease and death are lower the younger a person is, there still is a risk, and vaccine greatly reduces that risk. So it prevents you from contracting the virus very, very effectively. But most importantly, the only way we're going to stop this pandemic and stop the evolution of new variants that could potentially compromise so much of the effort that has gone into this vaccination is through vaccination. That we cannot rely on natural immunity to create enough immunity in our population to stop transmission. And it's really the ongoing transmission of the virus that is the big problem, because the at-risk individuals, which age is usually one of the greatest predictors of risk. But as you mentioned, there's other associated risk factors that can make an individual more at risk of disease and death. By having all of us vaccinated, those individuals are protected. And that's particularly important because some of those most at-risk people, the reason they're so at risk is that their immune system isn't quite as potent, is not as functional and able to fight off the virus. So it's better to just stop the virus transmitting in our population to protect those individuals. So we can't put all the responsibility on those who are most at risk, because yes, the vaccines work, but even now where we're hearing about, there will always be people who are fully vaccinated and will contract the virus and die tragically because the vaccines are just not 100%. The way we can make them 100% is to stop the pandemic by all of us getting vaccinated, because once we're all vaccinated and we stop transmission, then any sensitive individual or at-risk individual in our population can't be infected, because we've actually potentially driven the virus to extinction. That's the ultimate goal. And we have achieved that in the past with other infectious disease. And my great ideal and great wish would be that we would achieve that with COVID. It's going to be a hard road, and I doubt we're going to get there, but we get a lot closer, and we protect and save millions of lives by each of us getting that vaccine when we're able and when it's available.
Okay, and Janet again is saying, thank you for answering my questions, Dr. Cameron. This presentation has been very interesting. Keep up the excellent work you and your team are doing.
Thank you. We certainly will. We'll keep going always.
And that's it for the questions, unless they want to raise their hands. I got no more. Lisa?
No more questions, I guess?
No more questions.
Thank you, Hertha. Just want to say thank you again to everyone here today that's joined us and to your contribution to the conversation. I think the questions were excellent and they provided additional insight on top of the presentation that was done. I also want to thank you, Hertha, for facilitating the questions and helping us manage that in an orderly manner. And of course, Dr. Cameron, thank you for your time today and for sharing your knowledge and expertise with the community on the call today. The work that you're doing is absolutely critical to the success that our community and our country and the whole world has in beating this pandemic. So yes, I'd agree with Janet. Keep up the great work. Your team is doing a wonderful job and is helping us move forward and regaining some of the life that we remember before the pandemic. So hopefully we'll get to that point again soon with the assistance from science and scientists like you. A video of today's research with impact lecture will be posted on the alumni section of our website. And a link to that website can be found in the chat. Feel free to share the link with your family and friends. You can distribute the video as you'd like, and we'd love to get the message that Andrew is sharing with us today out to more people. And don't forget to check the University of Regina website. There will be more lectures, so we will be posting information on lectures in this series and future lectures in this series on our website. Our next and final lecture in the UofR Research with Impact series is on August 17th. And on August 17th, we will feature Dr. Tarun Katapalli, who is a physician and a patient-oriented research leader and associate professor in the Johnson Triama Graduate School of Public Policy on the UofR campus. And Tarun will speak on the future of public health, citizen science, and social innovation. So another interesting topic related to health. I hope that you can all join us on August 17th, but until then, please enjoy your summers, stay cool, and have a wonderful, wonderful day. And also please stay healthy. Take care.